All right, so this is the second video in our cell reproduction part 1 series here. In this section, we're going to take a look at mitosis and meiosis and go through some of the details related to each of those two events. So in the last part, we talked about the overview of the cell cycle. We talked about the organization of the chromosomes. We talked about how cell reproduction works in prokaryotes with binary fission and a number of other details there. So we're ready to talk about how in the cell cycle the chromosomes themselves are organized and how that relates to the actual division of the cell. So mitosis is defined as the division of the nucleus in which chromosomes are separated into two nuclei, sometimes referred to as the division of the chromosomes themselves. In this process, we see that the eukaryotic chromosomes are meticulously organized and separated so that each of the two daughter cells gets a distinct copy of each of the original DNA from the parent cell. So you can kind of see the overview here in the figure. Prophase, metaphase, anaphase, telophase, and cytokinesis all are parts of this mitotic cell division. Part of the necessity of mitosis is it ensures that each daughter cell gets an identical copy of each chromosome. So we're not just taking all the chromosomes and sort of grabbing two handfuls and splitting them apart. We're getting the exact replica of the parent cell and making sure each daughter cell gets an identical copy. We got a lot of DNA in the cell. Everything's been duplicated, so now there's double the amount of information. And mitosis ensures that each of the daughter cells get the exact copy that we had in the parent cell. It's a pretty amazing process, really. And what's going on is all related to that outcome of ensuring that we get identical copies in each of the daughter cells. So this term mitotic cell division is the process, it describes the process by which the eukaryotic cells undergo mitosis, followed by then cytokinesis, which is the process by which the cells split apart in the cell cycle. So different types of cell division may not include mitosis. So you have binary fission, which is a whole different process, but then we also have meiotic cell division, which we'll talk about later, which involves meiosis followed by cytokinesis. So this term mitotic cell division kind of differentiates that really from meiotic cell division, which is a different process. Now in single-celled eukaryotes, mitosis or meiotic cell division is the primary means by which many single-celled eukaryotes can asexually reproduce. So for example, in yeast cells, yeasts can simply duplicate their cell content and with mitosis split apart to make two yeast cells that are identical. In other words, they are copies of each other. So this is a form of asexual reproduction, simply copying from one to the next. So this is how some simple eukaryotes go through life, just simply copying themselves, making multiple variations with each new cell so that the population can be propagated. Now in backing up here real quick, we do get variation, I should say. Variation is, however, the result of mutation. So in general, mitosis makes exact copies, but those copies can vary due to random mutations that happen either from the environment or from errors in replication itself. So we don't always get the exact same thing, but mitosis aims to make an exact copy, even though Mother Nature tends to create variation in spite of that. But essentially, it's a means for asexual propagation in eukaryotes. Now, in multicellular eukaryotes, it doesn't work the same way. In multicellular eukaryotes, it's a primary means by which cells are generated during development. Now, we start out as a single-celled zygote, which we'll talk about later, which is the result of meiosis. Once we have a cell, we can start to copy that cell to make more cells with mitosis. But we're making copies to make more cells, not to make new species or new members of the population. So we're basically growing our organism by copying the starting cells to make more and more and more cells. As our bodies become developed and fully mature, this mechanism then becomes the primary way that we replace damaged or worn out cells in the body. So skin cells, I mentioned in the last video, have to be regenerated every day. Mitosis is the means by which we propagate copies of pre-existing cells to replace the worn out cells. So it can be used for development to create cells as we are starting out in life, and it can be used for regeneration of worn out or aged cells as we reach maturity later in life. So mitosis has about 5 stages overall. This does vary depending on the source, but those 5 stages are prophase, prometaphase, metaphase, anaphase, and telophase. Now, one little note, as I mentioned here, prometaphase is often not listed in many sources. What you're doing is you're sort of taking this process and you're sort of arbitrarily dividing it up in different stages. And so what a lot of people do is they just sort of go from prophase to metaphase. Prometaphase in between there is a more technical, nuanced stage that isn't always listed. So be aware of that if you're looking at different sources. Oftentimes you just go from prophase to metaphase and then anaphase and telophase. But here I wanted to talk about prometaphase because it is listed in some sources, and so it's better to give a little bit more information to be more thorough than to leave out that information. So don't try to let that get you confused. It's really the same process. It's just whether or not you want to identify that extra little detail in between those first and third steps there, and a lot of times people don't. But we are going to here. So we're going to discuss each stage here as we go forward. So starting with prophase, pro means first, and prophase is the first phase that was able to be identified with some of the earliest microscopes that were available to scientists. So the way it's often described is prophase is the stage in which the chromosomes condense and actually become visible with a light microscope, to say. Now there's a lot more happening here. During this stage, a structure called the centrosome produces microtubules, these little spindle-like structures that we see here. They're kind of like little arms or tentacles, and they radiate out from the poles of the cell towards the chromosomes here in the middle. Now, in the next stages, we'll see what those start to do more and more. Initially, though, the centrosomes are producing these little arms, these little tentacles that will branch out and grab a hold of the chromosomes. And in prophase, those centrosomes are just being produced. Now, here's a better figure of what that kind of looks like. The centrosomes are actually made up of individual centrioles. I know that's very confusing, but the centrosomes, as it's shown here, are the collective structure, this whole thing here. Individually inside here we have individual what are called centrioles. These are the smaller units of the centrosomes themselves. I know that's confusing, I apologize. I'm not going to try and trip you up on that later, but they're named similar because they have, they're all part of the same feature. So the centrosome is the unit, the centrioles are the individual parts of that unit. What the main thing I want to get across here is they do this really amazing thing, which is they produce these little structures called microtubules, sometimes called spindle fibers, as they're listed here, astral spindle fibers. It's basically an array of little tentacles that reach out from each side of the cell. So these are produced on both ends of the cell. They reach out towards the middle, and they'll actually grab a hold of the chromosomes, and this is the means by which chromosomes are moved around the cell. So as we'll see here in the next stages, these chromosomes start to move, they become aligned, they move from one side to the other, and the way that happens is with these little tentacles, these fibers that branch out from these centrosomes here and grab a hold, and they push and pull in both directions out. So going back here just real quick then, this is what begins to form. The centrosome and the centrioles begin to form during prophase. And they'll tend to start to migrate to both sides of the cell, or both poles of the cell at the same time. In prometaphase, what is technically happening here is the nucleus starts to break down, and these microtubules, or these spindle fibers, will actually start to attach to the individual chromosomes themselves. So as I mentioned, these little fibers are going to be moving the chromosomes around. So in prometaphase, they'll attach to the chromosome, which is obviously a prerequisite for them to be able to move them. Specifically, the microtubules, or the spindle fibers, attach at areas called kinetochores. Kinetochores, looking at this figure here, kinetochores listed right here, are located on each side of the centromeres, which we talked about in the last video. So centromeres are what hold together sister chromatids. So here we have two sister chromatids. These are duplicated copies of the same chromosome, and they're held together by the centromere here in the middle. Kinetochores are located on each side of that, look like little lima beans here, but these kinetochores are the attachment sites for the spindle, or the microtubules, that attach to each side of these duplicated sister chromatids. So during prometaphase, the spindles, the microtubules, attach to these sister chromatids at the kinetochore. So each kinetochore is associated with one of the two sister chromatids, and what this means then is that each of these sister chromatids will be connected to its own microtubule, or its own spindle fiber. I'm calling them different things here because different sources use different terms. So they're actually what are made up of microtubules, and in this context with mitosis we can call them spindle fibers or astral spindle fibers, and so we get a lot of different names for them, but these little structures that connect to the chromosomes here. Now it's important to note that they're connected to each of the sister chromatids, and so what's going to happen is, we'll see in the latter stages, is they're going to be pulled apart as these microtubules pull in opposite directions. These sister chromatids are actually going to be broken apart later. But for prometaphase, they're attached, and that's really what's the main thing happening in prometaphase. So this becomes essential later, as I mentioned. Now in metaphase, what's going to happen is those microtubules, or those spindle fibers, will actually push the chromosomes until they're aligned down the middle of the cell. So meta means middle. And what's happening is the chromosomes are actually being lined up in the middle of the cell. Kind of an amazing thing. These have sort of an uncanny ability to innately line up these chromosomes right down the middle of the cell. So one of the most obvious stages of mitosis is called metaphase, because you can see it nice and easily because all the chromosomes line up right in the middle of the cell. And that's really the main thing about metaphase. The next phase then is anaphase. In anaphase, the sister chromatids will actually break apart as that centromere that held them together breaks down. So this is going back to that figure I showed you there. That centromere is what holds them together. In anaphase, it's going to be broken, and the two sister chromatids can now be pulled in opposite directions, and this is going to happen as the microtubules, or the spindle fibers, start to contract and shorten, pulling each chromatid in opposite directions. So you can start to see what's happening there. Those fibers got a hold of them, and they're pulling them towards opposite directions there. Here's another figure showing kind of the same thing there. So this releases the sister chromatids, and they move in opposite directions towards the poles of each of the cells, each side of the cell. And this is where the split will come later, and we'll see that each side will become its own daughter cell at the end of this process. So this is really when the duplicated DNA splits apart, and each side is going to start to get its own full complement of the original DNA from the parent cell. Telophase is probably one of the more complex stages. There's a lot going on in telophase, and so we're kind of summarizing quite a bit here. But what happens in telophase is the nuclear envelope starts to reform, and the nuclear envelope that was initially broken down starts to reform around the nucleus and the nuclear material on each side. So the chromosomes have reached opposite ends of the cell. That's one of the first things about telophase. And the nuclear envelope begins to reform. Sorry, I went out of order here. The nuclear envelope begins to reform, and the chromosomes begin to recondense back into their original form. So initially, the chromosomes condensed. They were in a loose form. They condensed early in the process in prophase, and now in telophase, they're decondensing back into their loose form. And at that same time, the nucleus is reforming around each side, And at that same moment, the cell is starting to pinch apart. It's going to start to divide down the middle, which will fully complete in the next stage here. So to recap telophase, the chromosomes have reached each side of the cell. The chromosomes begin to decondense back into a loose form, and the nucleus begins to reform around each side of these newly forming daughter cells here. The last stage is cytokinesis, and cytokinesis stands for a division of the cytoplasm, and it's basically when the cell actually splits. So it's the last stage. The cell will split in two, and it's a little bit different depending on the types of cells. And so most textbooks will kind of summarize this as how it's different between animal cells versus plant cells. So here I've got some figures giving you examples of each here. And animal cells, they're nice and squishy and kind of flexible. And that means that they can be more easily pinched apart. And what you have is the formation of a contractile ring, a lot like the Z ring that we talked about in the last video. And it basically is a protein band that pinches as it contracts. It pinches the two cells apart as it contracts down the middle. And as it reaches the last section, it pinches the two cells apart from each other. In plant cells are much more rigid, and they are more like cement walls with each cell, whereas animal cells are more like squishy, more like a squishy sack of fluid. So plant cells, you can't pinch them apart because they're just too rigid for that. They'll break apart before they pinch. So the way that works in plant cells is the plant cells grow, and then they actually synthesize a new wall in between them. And this is called the cell plate. The cell plate is actually what starts out and then ultimately becomes a new cell wall later. So basically in animal cells they pinch apart, and in plant cells they grow to divide by creating a cell wall in between them, kind of dividing up the cell that way. Either way, you have two new cells formed from the duplication of the original parent cell. Following cytokinesis, the cells are now free to start functioning again. Many cells will go right back into that process and begin the process all over again, starting with the G1 phase. As I talked about before, some cells divide rapidly, they pretty much never stop. Other cells divide very slowly, and many cells are kind of in between that spectrum between fast and slow. But the idea is that they can now, depending on the cell, depending on the circumstances, either go back into division or go back into normal functioning. So next I want to talk about meiosis, and I'm going to start by contrasting that a little bit with mitosis so we get a better idea of how these two processes are different. So mitosis and meiosis are two things that we want to get straight here as we talk about them. So mitosis is what occurs in what are called somatic cells, also known as body cells. Somatic cells are basically all the cells in your body except for your sex cells. Sex cells are called gametes or gametes, and these include the sperm and the egg, which are really specific cells that are produced in the ovaries of women and in the testes of men. So mitosis is what occurs in all the other cells except for these gametes. Meiosis is what occurs in the gametes, the sperm and the egg, or I should say it's what occurs in order to create these cells, is the best way to put it. So there are cells that undergo meiosis in order to create sperm and egg. So these cells undergo what is called a meiotic cell division as opposed to a mitotic cell division. So it's similar to mitosis or the meiotic cell division, but there is significant differences in between the two, and the differences create a very different outcome at the end. So it's interesting because the outcome of meiosis is really completely different than mitosis, but the processes are actually similar. But as I'll point out here, there are a couple of key differences along the way that create the vast difference in outcome at the very end. One of the main differences, mitosis goes through a single round of division. One cell splits to become two cells. Meiosis goes through 2 rounds of division, where one cell splits twice to become 1, 2, 3, 4 cells. So that's one of the main differences right there. How we get to that, then there's more detail in between there. So we talked about the difference between diploid and haploid in the last video. diploid organisms have two sets of their DNA, or I should say diploid cells have two sets of DNA. Haploid cells have only one. With meiosis, what we're actually doing is we're creating 4 haploid daughter cells. These will be the sperm and the egg. So the haploid daughter cells are the sperm cells or the egg cells, whether you're male or female. And those cells are actually haploid. They're actually the only haploid cells in your body are sperm and the egg. All the other cells in your body are diploid. So this is where meiosis creates that difference. We have haploid versus diploid cells. Now, the reason that you do that, it makes perfect sense when you stop and think about it. We know what sperm and egg do. They combine during sexual reproduction to create an offspring. Specifically, this is a process of fertilization, and fertilization gives rise to the single cell that we all start out in life as, which is called a zygote. And a zygote, as a human being, a human zygote that is, will have exactly 46 chromosomes, 46 total chromosomes. Now, the sperm and the egg themselves will each carry 23 chromosomes. Being haploid, they have 23 as opposed to 46 chromosomes. And the way that works is when the sperm and the egg combine, they two add their DNA together to give us the 46 overall chromosomes, which then go on to develop into the embryo, into the fetus, and then into the human being itself. But that process begins with 2 halves creating one whole. The reason we do it that way is because sexual reproduction allows DNA from different members of the population to join together to create a completely different member of the population that is genetically distinct from all of the members of the population. So you as a human being are a genetically distinct individual that is unlike any other human being that has ever lived and ever will live. And that's because the DNA from your parents was combined in a completely unique way to create you, which makes you a unique individual, as with every other member of the entire population in the world. We're all genetically distinct. We're similar, but distinct. And part of the idea of sexual reproduction is we create that variation, which gives our population lots and lots and lots of differences to make us better at carrying out different tasks, surviving different types of diseases, being more fit for different types of environments. So variation is one of the key hallmarks of survival over the long term in evolution. I talked about that a little bit at the end of the last video. Sexual reproduction is one of the main drivers of that diversity. We combine different sets of DNA to create new, unique offspring, as opposed to just copying the DNA over and making the same version that we had. So this is why sexual reproduction is so different. And this is also why we need to create haploid cells, so that each cell carries half the information to combine to create the whole. So as I said, the result is that you're a unique individual rather than an exact copy. So recapping what I said there, the difference is that the result is a genetically distinct individual rather than identical copies. This increases the genetic variation of each cell. And in the grand scheme of things, this is important for the evolution and survival of a species. The more variation that we have, the better we can adapt, the better we can change, the more likely it is that some members of the population will survive, even if not all members of the population do. Right now, we're dealing with the coronavirus outbreak. One of the things that's interesting about virus susceptibility is that based upon your genetic makeup, you can be more or less susceptible to different kinds of viruses. Now, we're still studying the coronavirus, but people can be more or less genetically susceptible to different kinds of viruses. So a virus is not likely to ever take out every single member of the entire population, because every member can be slightly different enough to where you and I might have a different immunological advantage to fight different kinds of viruses. So that's just one example of where adaptation and variation can be significantly important. With viruses going around, not everybody is equally susceptible. And that's partly based on your health, your overall health, and how much sleep you get and how much food, the nutrition you get and all that kind of stuff. But it's also based upon the actual makeup of your DNA itself. So genetic immunology is part of what makes us more or less susceptible to viruses, in addition to how well we take care of ourselves. So that variation helps us survive things like that as a population. Even if not every member even if lots of the population dies, some members of that population will continue to survive, almost no matter what. If we were all identical, a single virus could theoretically take us all out, and the whole population might be erased. So that diversity is key in long-term survival. Okay, next I want to look at meiosis. The overall, we can take a look at the overall process of meiosis. So in some ways, it's similar to mitosis. In the theory of evolution, it's believed that meiosis evolved from mitosis. In other words, mitosis came first, and then meiosis was an evolutionary branch that extended out from that process. In other words, this is similar because it's thought to be evolutionarily related. One of the main differences is that we have 2 rounds of division, like I said. So rather than just one cell splitting once to make 2 cells, that is how it starts out. With meiosis 1, we go through all the same phases: prophase, prometaphase, metaphase, anaphase, telophase. And then in meiosis 2, the 2 daughter cells that are created at the end of that process go through that process each individually all over again. And those cells will end up creating 4 genetically distinct cells at the end. So we go through 2 rounds of division to create 4 haploid daughter cells. Now, many of the events are the same. You have the same concept in which the cell prepares for division with interphase. The DNA is duplicated, the chromosomes condense, the spindle fibers are produced, and really a lot of the same things are happening, as I mentioned. But there's a couple unique events in meiosis that differentiate the overall outcome and make everything significantly different by the time we're done here. So rather than go through every single step, what I want to do is kind of just focus on what those unique events are. So the first one is that early in the process, in meiosis 1, there's a unique event in which there's a pairing of homologous chromosomes. So that pairing of homologous chromosomes means that those matching sets of chromosomes are actually going to all join together to make what is referred to as a tetrad or a bivalent. Once that tetrad or bivalent forms, we'll then have a process called crossing over that occurs within those that makes each set of homologous chromosomes genetically distinct. And then finally, another key event is that we have a reduction in chromosome number. And this is what allows us to go from being diploid to being haploid. So pairing of homologous chromosomes, during prophase 1 of meiosis, homologous chromosomes will pair with each other. They form what is called a tetrad, also known as a bivalent, depending on what textbook you're reading or what source. So here's what that looks like. Initially, we have 2 sets of homologous chromosomes. So these would be the matching set, call it chromosome number 1, for example. And we've got the duplicated chromosomes, so we have 2 copies now after duplication. So we've got 2 sister chromatids here and 2 sister chromatids here, and these are the homologous chromosomes that are the 2 sets from each parent. So the blue might be the chromosomes from your dad, and the red, the chromosomes from your mom, and they've both been duplicated, so we have 2 sets of each now. Now what's going to happen early in prophase 1 of meiosis is they're going to join together and they're going to fuse to make what's referred to as a tetrad or a bivalent. This does not happen in mitosis. In mitosis, the homologous chromosomes effectively just act independently. They don't associate together. They're not in any way combined at all. Even though they're the same type of chromosome, they act independently completely. In meiosis, however, they somehow find a way, it's one of the miracles and mysteries of life, they somehow find a way to connect to each other, and they form what is referred to as this tetrad or this bivalent. Now, this process is referred to as synapsis. So synapsis is the process by which these matching homologous chromosomes effectively fuse together, at least temporarily, to form this tetrad. Again, does not occur in mitosis, and it is one of the most significant differences in the overall events between mitosis and meiosis. A little bit out of order. Go ahead and put these up here. The next thing that's going to happen after the formation of the tetrad or the bivalent is called crossing over. So once that tetrad forms via synapses, so synapsis is the process that leads to the formation of the tetrad. Once that forms, there are these spots along the tetrad called chiasmata, where the two homologous chromosomes will effectively fuse together. So they're like little areas where the two chromosomes basically stick together. These sections are called chiasmata. In the majority of those chiasmatic locations, you have a process called crossing over that takes place. And crossing over is where a little piece of one chromosome will actually break off and fuse to the other chromosome and vice versa. So they effectively swap genes between the two homologous chromosomes. So these homologous chromosomes carry the same types of genes, but they can be different variations of those genes. So in other words, they serve the same function, but they may be different in terms of their variability. They may not be the exact same gene. So by switching these up in little sections, it means that each of these chromosomes now is genetically distinct. We started off here with all solid dark blue, all solid light blue here, just based on these colors, and we end up with recombinations of those two different chromosomes being pieced together to create a new, unique variation. And what this means is that these resulting chromosomes that are the result of crossing over are genetically distinct from the parents. They still carry the same genetic information, but they don't carry it in the same combination. So you can have different combinations of unique genes that mean that the offspring now will automatically be different from the parent. Even before sexual reproduction takes place, the DNA of the offspring has already been different from the parent. The parent DNA looks like this, the offspring DNA will start to look like this, and that is going to be automatically different from the parent. So that it's going to create variation. And again, going back to the evolutionary theme, this is one of the ways that we get unique variations, increase that variation really, in our offspring. So this is called crossing over. It's a very interesting, weird little thing, but chromosome segments swap, and that leads to variation. That's the main takeaway there. The next key event is the reduction in chromosome number. So in meiosis, we end up with haploid daughter cells, as I mentioned before. This means that somewhere along the process, we lose half of the genetic content, specifically 1/2 of the homologous chromosomes. This is where they go from diploid to haploid. In humans, for example, our cells go from 46 to 23 chromosomes, as we showed earlier. So they still have all the genes for functionality, they just don't have the extra set of DNA. So each sperm and each egg technically carry all the information you need, they just don't have two copies of it. And so each of those will carry 1/2 of the overall total. So this is, I mentioned this already, but this is what we're going to just describe a little bit more about how this actually happens here. So this is where the reduction in chromosome number. So this process actually begins in prometaphase 1 of meiosis. So if you think back to what I talked about in mitosis just not too long ago, in prometaphase of mitosis, this is where the spindle fibers attach to the chromosomes. What's different here is the microtubules don't connect at the exact same place that they connected before. They connect in a slightly different location to the kinetochores, as opposed to they connect to kinetochores that are fused together between each of these sister chromatids, as opposed to each side of the sister chromatid. I'm going to give you a better figure here in just a second. But what they're doing, what's key here, is that the spindle fibers will attach to both of these as a unit. So one set of spindle fibers attaches to this pair of sister chromatids, the other attached to the other pair of sister chromatids. And what's going to happen is when they pull apart, they're going to pull both of these chromatids in one direction and the other chromatids in the other direction. And what's going to happen is we're going to end up losing one set or the other in each of the daughter cells. So here's a better figure to kind of show how this is different between meiosis 1 and mitosis. So this figure here does a little better job of showing us side by side how this is different in meiosis 1 versus mitosis. So in meiosis 1, these sister chromatids are grabbed as a unit. So one set of fibers comes in here and grabs this set, another set of fibers comes in here and grabs this set, and as they pull them apart, they take one set or the other to each side. In mitosis, what ends up happening is each set of spindle fibers only grabs one side of the sister chromatid or the other. So the sister chromatids are separated, so each side of the cell will get both of the sister chromatids. In meiosis, each side of the cell will get one set of sister chromatids or the other, but not both. So this is where we end up going from diploid to haploid, because in order to be diploid, we actually need both sets of homologous chromosomes. In this case, we need the red and the blue. In mitosis, we separate so that we get both red and the blue. In meiosis, we separate so that we get only the blue or the red, but not both. And so what this means is that we end up going from diploid to haploid. Now, I know this is probably not making sense just yet. So what I did was I put together an extra little video where I demonstrate this with some beads in the lab. So I'm going to insert that video now so you can watch that video, and hopefully that'll make a lot more sense so you can see better on what I'm talking about. And when that video is finished, then I'll come back and we'll start from where we left off here. I want to use these beads as a model to represent chromosomes to kind of show you what the differences are between mitosis and meiosis I. So what I have here is a hypothetical eukaryote with 3 chromosomes that is diploid. So it has 3 chromosomes with 2 pairs of each chromosome, each pair represented by a different color. So blue and green are one pair. These are homologous chromosomes. Red and yellow, purple and orange. These are homologous pairs of each chromosome. Now, one of the things that happens that's the same between mitosis and meiosis I is just before those, we have duplication of the chromosomes. So each of these chromosomes is duplicated, and that is now represented here by these extra set of beads that are being added here. So prior to mitosis and prior to meiosis I, we have a duplication of the chromosomes, meaning we now have 2 copies of each of those chromosomes. Now, what's going to happen in mitosis is that all of the chromosomes are going to line up at the metaphase plate independently like this. So each of these represent a pair of sister chromatids. The 2 blues, the 2 greens, the reds, these are sister chromatids. And those sister chromatids are all going to line up independently at the metaphase plate during mitosis. And during anaphase, then, each of these is going to split and move to their respective sides like this. This extra piece of tape here represents what then happens in telophase, which is these 2 split apart to make their own separate cells. So this is the end of mitosis. We now have 2 daughter cells resulting from the split here. Now, if you look, what we have are the exact same situation that we started with, meaning that all the chromosomes we had in the parent cell are now found in each of the daughter cells. And that's because we duplicated them and evenly split them apart in mitosis. Now, if we go back and look at what's going to happen in meiosis, one of the first things that's going to happen differently is that these homologous chromosomes are going to pair up in a process called synapses. So what that means is that in this case, the blue and the green chromosomes, those are going to form a tetrad complex, or sometimes called a bivalent complex, which occurs with synapses, where all these chromosomes basically fuse together into one unit called the tetrad or the bivalent. And that's going to happen for each of the homologous chromosomes like you see here. So these all form what we now have 3 bivalent or tetrad complexes. The next unique event of meiosis is crossing over. So now that we have these complexes set up, what we'll now see is that little pieces of the closest touching sister chromatids here are going to actually swap genetic elements between their homolog next to them. So for example, a little piece of the blue chromosome will come off and swap with the green like this. So we now have slight variations in each of these chromatids here in the middle, where they're not completely made up of the blue chromosomes, they now have slight mixes between the green and the blue. And that's going to happen on all 3 of the chromosomes. So now when you look, we notice that each of these chromatids here in the middle have a different variation and a different makeup of genes that come from each of the 2 different homologous chromosomes. So these can be the same genes, but they can be variations of those of the same gene. For example, if we have a color, a gene that codes for pigment, the genes both code for pigment, but they might code for a different pigment. So a black pigment versus a white pigment, for example. So we might get different color variations, different size variations, things like that. Now, that's the next thing in meiosis. So we have the synapses crossing over. The next thing then is the way that these tetrads or bivalents line up at the metaphase plate in meiosis 1. So this would be metaphase 1. What's going to happen is the entire tetrad or bivalent complex is going to line up like this at the metaphase plate. Now, during anaphase 1, when these go to separate, what's going to happen is they're going to separate as homologous pairs, or as sister chromatids like this, where each of the daughter cells is going to get one set of homologous chromosomes or the other, but not both. So in mitosis, each of the daughter cells gets a green and a blue. Now we've got each of the daughter cells getting either the green or the blue, with the crossing over that's taken place being the only exception there. So we now have, at the end of meiosis 1, telophase 1 coming in, splitting those 2 cells apart. At the end of meiosis 1, we have 2 daughter cells, but now each of these daughter cells is now considered to be haploid. And they're haploid because they only have one set of the homologous chromosomes or the other, and not both. In order to be diploid, I need 2 sets of each chromosome. For example, I need a green and a blue. I'm haploid if I only have one set or the other. And that's exactly what's happened here in the end of meiosis 1. Now I have 2 copies of the green and 2 copies of the blue, but it's not the number that makes the difference, it's the quality. So I have only one of the green or the blue and not both. And that's what makes me haploid in this situation. Now in meiosis 2, what's going to happen is the remaining DNA on each of these daughter cells is going to divide again. And this is going to look a lot more like mitosis. So what's going to happen here is these are going to line up at the equator. We'll do it this way this time. Here's the equator here in the middle. So this is going to line up like this here. I'm running out of room here. But we're going to see them line up like we did in mitosis. So the same thing will take place on the other side over here. And now, as they go to divide, what's going to happen is each of the daughter cells is going to get one of these 2 sister chromatids that is remaining. Like this here. And our dividing line will now show that we have 1, 2, 3, 4 daughter cells at the end of meiosis 2. Now notice that each of these daughter cells has only one of the 2 original sets of chromosomes. So they are now haploid. We have only one blue chromosome, the purple here. Now the difference is that we have elements of the other chromosomes sprinkled in there. So I have parts of the orange and parts of the purple in at least some of these. And so each of these daughter cells not only has haploid condition, but they also have a mix of DNA that was different from the original parent cell. So each of these now has a unique combination. If you look, each of these chromosomes, the combination of chromosomes is different for each daughter cell. So this daughter cell has the original red, blue, and purple, but this daughter over here has the original orange and yellow, but a recombination of the green and the blue. And this one down here has recombinations of all 3 chromosomes. This one up here has recombinations of 2 chromosomes and one of the original. So every daughter cell is genetically distinct. Now these are slight variations. These are not like completely different species or completely different sets of DNA. They're slight variations, but they are variations. And in nature, over the grand scheme of things, those slight variations can actually make a big difference. Each of these daughter cells will contribute a slightly unique set of DNA to the future offspring, and that ensures that the future offspring will be slightly different from the parent. And that means that over long periods of time, our population has the ability to change slowly and adapt to new potential environmental situations. And that's what makes meiosis so unique and so different, is that it creates variations, and it also reduces the overall chromosome condition from diploid down to haploid. Okay, so hopefully that helped you understand what I was talking about a little bit better there. You can see the reduction in the chromosome number that results because we're pulling only one set of those homologous chromosomes and not both. Now, the other thing that makes each daughter cell more diverse is the fact that each time we pull homologous chromosomes, they can be pulled in different directions. So here's an example a figure that shows that. In one set of scenarios, we see that the blue chromosomes and the red chromosomes line up on each side, and as the cells divide, the daughter cells get either blue or red and split to create these unique daughter cells down here. And another possibility that's equally likely is that instead of the blue and the red lining up on opposite sides, we get one blue and one red lining up like this, so that when they split, we get blue and red on one side and red and blue on the other, and each daughter cell then gets a more unique combination of each of those different homologous chromosomes. So this is what's referred to as random orientation, or sometimes called random assortment. What it basically means is that each time the chromosomes line up, it's basically a coin flip as to which side each chromosome lines up on. So in one scenario, we get blue and blue and red, and in the next scenario, we get blue and red and blue. So it's not always the same in how they line up, and then as they're pulled apart, we get different combinations of the homologous chromosomes, making each daughter cell even more unique. So this means that each set of homologous chromosomes can be mixed up differently with each new daughter cell, meaning that we get even more variation than we had with crossing over alone. So this is what's again referred to as random assortment or random orientation. This leads to unique combinations with each given daughter cell. So here's a video from McGraw-Hill, one of the textbook publishers. I'm not going to play it here because I might get my video pulled, but it's not great, but it gives you a little bit better idea of what random orientation looks like. You can find this on YouTube. Just type in random orientation of chromosomes during meiosis, HD animation. This will probably be the first one that comes up. And what it basically shows you is how these different chromosomes can line up and orientate differently, and it goes through some of the numbers of how that can create lots of different variations. So I understand this can be kind of a confusing topic. It's hard to find good visuals for this, which is kind of frustrating for me. But random orientation means that basically each of the homologs can line up differently, randomly, and we can get different combinations in the daughter cells as they split apart during anaphase. So once the chromosomes split apart in anaphase 1, they'll go through telophase 1 in the exact same way that they went through telophase in mitosis. The DNA will, well, not the exact same way, it's very similar. The DNA doesn't fully decondense like it does. That's one of the main differences. The nuclei don't fully reform. It's another difference, but it's very similar. So with meiosis 1, we have pretty much all the major differences that occur, where we have homologous pairing, crossing over, a reduction of chromosome number, and the random orientation to make the genetic diversity even greater in the daughter cell. At the end of meiosis 1, what we end up with is two daughter cells that are each now haploid, but they still have two copies of each chromatid. So we only have one or the other homolog, so we're no longer diploid, but we still have two copies of each of those individual sister chromatids. So what's going to happen in meiosis 2 is the remaining DNA of each of the remaining copies is going to be separated again, and we're going to end up with only one of the two copies in the last of the four remaining daughter cells that are produced at the end of the process. So meiosis 1 ends with telophase 1. The chromosomes uncoil slightly. The nuclear envelope doesn't fully reappear, maybe slightly. And ultimately, telophase 1 kind of blends into the next part of meiosis 2, which then begins with prophase 2. Now, in between meiosis 1 and meiosis 2, there is no interphase. We don't go back into interphase. That means that there is no replication of the DNA in between meiosis 1 and meiosis 2. So that's a significant thing to point out there. The result is that we'll only end up with 23 chromosomes when we're done. If you were to replicate your DNA in between meiosis 1 and 2, that would mean that you would go back to having double the amount of DNA, and when the daughter cells were finished, they would have 46 chromosomes rather than 23. So we don't want to duplicate the DNA in between meiosis 1 and meiosis 2. This ensures that the daughter cells at the end will only have 23 chromosomes, which, as we've already discussed, is what we want. Replicating DNA in between those would lead to a double in the number of chromosomes, and this is something that Mother Nature just does not allow for. The chromosome number is very specific, and it has to be maintained in order for the zygote, or the sperm and the egg themselves, to be viable. So it's very important to note that meiosis, interphase is not occurring in between meiosis 1 and meiosis 2. Now, once we get to meiosis 2, we now have 2 daughter cells resulting from meiosis 1. And at this point, meiosis 2 looks a lot more like mitosis itself. In other words, the chromosomes will line up independently and split apart without anything extra going on. There's no more pairing of homologs, there's no more crossing over, there's no more reductive division, there's nothing like that. So basically, it looks a lot like mitosis, in which things line up and they split apart. And what's happening is we're splitting the last 2 pairs of the chromosomes that we have left after the result of meiosis 1. So starting with prophase 2, the major difference here is that these daughter cells are now haploid and not diploid, but they still have 46 chromosomes. They just don't have the 2 pairs of homologous chromosomes. So, but what's going to happen, they got 46 chromosomes, and we're going to split those into 23 in the remaining daughter cells down here at the bottom. So we'll have a total of 4 haploid daughter cells at the end of meiosis 2. Here's a quick summary table. I'll leave this up here if you want to pause that. You can go back and look at this. This gives you some comparison between mitosis and meiosis and some of the different distinct differences that occur between them. And just to summarize, meiosis is the basis of sexual reproduction. Sexual reproduction itself actually involves 2 separate processes. Meiotic cell division is one part, but then fertilization is the other, so in which the sperm and the egg are combined. So this is where meiotic cell division creates the sperm and the egg, and then fertilization will lead to the formation of the zygote itself. The zygote now is diploid, having 2 complete sets of chromosomes, 1 from each parent. So meiosis basically gives us those haploid gametes, the sperm and the egg, by reducing the chromosome number down and also creating variation, so each of those sperm and the egg are going to be different from each other. And then when they combine in sexual reproduction, we get even more variation because the sperm and the egg were already varied, and when they combine with a new individual from a different partner, their variation combines with your variation to create a completely unique individual. So every individual on Earth is genetically distinct, even brothers and sisters, unless you're an identical twin. I should point that out. The exception is with identical twins. Unless you're an identical twin, then your DNA is distinct from all other members on Earth, all other people on Earth. And that's because meiosis does so much to create variation, and then the fertilization itself, combining 2 different sources, means we have that much more variation on top of it. Okay, so I know meiosis is definitely a confusing topic. You may have to go back over that. There's a number of great videos you can find online that give you the animations. I would recommend that you go find some of those. McGraw-Hill is a good one. They've got lots of short animations that help you see the process. I can't put those in my video because I get those pulled down, but you can go find those, and I would recommend watching those. So to summarize though, eukaryotes have homologous chromosomes. Karyotyping allows scientists to visualize chromosomes. That was actually from the last video there, that got misplaced. But mitosis produces identical copies. Meiosis is the basis of sexual reproduction. We have unique events in meiosis. And ultimately, those are the main things we wanted to summarize there. Now, in the next part, we'll talk about regulation of the cell cycle. So we'll do that in our next video series. We'll talk about how the cell controls cell growth, and this actually gets into how cancer starts and how the cells know when to grow and when to stop growing and things like that. So we'll look at that in our next video series. Until next time.